Welcome to the pan-genomic model, part 5. In the previous section, I concluded that transcription takes place in factories containing the appropriate transcription factors. For example, red promoters were transcribed in red factories and green ones in green factories. Today, I'd like to describe how various regulatory motifs all work. But first, a brief review of the transcription cycle. We imagine that a promoter binds to a polymerase in the factory, and then the transiently fixed polymerase extrudes the promoter as the transcript elongates. And it terminates by detaching from the factory. The important result in regulation, I believe, is the distance effect. So intuition tells us that D is likely to visit the factory and so initiate much more often than E. So there it's binding, initiating. And this is confirmed by simulations and experiments. Close tethering also ensures bursty transcription, because when this terminates, we go back to the structure, and then if D remains closely tethered to the factory, it's likely to reinitiate again, and we might go through a number of cycles. But if the polymerase at the top now detaches, D might diffuse away from the factory, and so the burst will end. Regulatory motifs, and in particular enhancers, which were originally defined as motifs increasing transcription independently of orientation or distance. Subsequently, they've been defined in many different ways. That's an indication of the fact that people don't really understand how they work. We will use a phantom definition, as I've described earlier, that says that they're just promoters that are active. And I would like to reinforce the idea that enhancer-promoter contact requires an active polymerase. And that's a quotation from the title of a recent paper. So D is initiating more often than E because it's closer, but then ask the question, why? Because of C. It's tethering D close to that factory. So if we focus on C, we would call it an enhancer of D, but it's just an active promoter. A superenhancer now, originally defined as sets of adjacent enhancers, in this case A, B, and C C is being transcribed at the moment, tethering D close. But when C terminates, we get a structure like this. But now we've got 3 times the chances that A or B or C might rebind to retether D close to the factory. And so it can reinitiate. So if we focus on A, B, and C, we would call them a superenhancer of D. Now to a silencer. And the first one turned out to be a tRNA promoter. Of course, we now know that tRNAs are transcribed in polymerase III factories. So F and D are tethered equally far from that pink factory. But if F visits the factory, it's unlikely to initiate there because green factors are missing. It would like to get across to that green factory, but it can't. If we focus on G, we would say it's tethering F close to the pink factory and so silencing it by preventing it from getting across. It's again just a promoter. Now, EQTLs. Imagine that we introduce into G a SNP that reduces the binding to the pink factory. So that enables F to visit the green factory and increase its firing frequency. If we now focus on G, we would say that the SNP is an up-regulating EQTL of F. It's just a promoter. So EQTLs can act transcriptionally, and not just post-transcriptionally, as in the omnigenic model. And that is why they often make molecular contact with their target genes. And by extension, I'm going to say that QTLs can also act similarly at the transcriptional level. Insulators and boundaries. The most famous insulator was originally defined as a TF, hence the TF in CCTF. Others turn out to be very strong promoters of tRNA genes. Boundaries, people have mapped what they are. They're often polymerases stalled at Hox genes. They often contain CTCF and polymerases. And in a beautiful paper called A Bacteria, boundary strength was shown to be directly related to the transcription rate of the promoter at the boundary, with transcript length determining the strength. And that paper also ruled out many other possible interpretations of how they work. So I'm going to treat these two together and imagine that C and B are being highly transcribed in that green factory. But of course, that factory contains a cloud of surrounding loops. And that cloud will prevent A from often visiting the pink factory on the left. And if we focus on B and C, we would say that they act as insulators or boundaries between G and A, preventing the two from contacting each other often. What I'm really saying is that any promoter can be considered an enhancer, a silencer, an EQTL, a QTL, or a boundary, and so on, depending on which target is considered, and that is why they share so many molecular marks. But they're all just active promoters, and they can act as one or other motif all at once. Now let's turn to the regulatory motif that was the first to be uncovered, heterochromatin in the fruit fly. So let's imagine that a green factory is made heterochromatic. That will change the frequency that all of the promoters are able to access factories in their surroundings. If we introduce extra euchromatic blocks with lots of active promoters, of course this adds more factories, which can act to stimulate or repress the activity of surrounding promoters. Similarly, if we introduce loops anchored by cohesin, and simulations show all these effects change the structure and the function both near and far away through these distance effects. And so they affect firing frequencies. And I find it difficult to predict whether promoters are going to be up or down regulated by such changes. Now, thus far, I've focused on interactions between promoters within a chromosome. In other words, cis contacts. Trans contacts, contacts with other chromosomes, are discarded from high-C data. And for that reason, trans contacts have been considered less important. And as a result, interactions between different chromosomes have not often been considered. However, they do occur. A promoter doesn't mind whether the pink factory is on one chromosome or another chromosome, or even on its homologous chromosome. And when the two initiate, the two chromosomes transiently become tethered together. So this enables us to provide simple models for things like transvection and mitotic and meiotic pairing. Now, what I'd like to do is to repeat really what I've said in a slightly different way. This pink factory might transcribe just estrogen response genes. This orange factory might transcribe housekeeping genes. If we focus on this grey promoter, it might visit one or other of these two grey factories to give different sets of 3C contacts. Two of the three green promoters associated with this factory might terminate, and that would allow that grey promoter to access the grey factory and so fire. If we consider this yellow promoter, it's simultaneously a silencer of red. It's just been an enhancer of the yellow promoter that is now being transcribed in the yellow factory. Now, perhaps this pink factory disappears and becomes heterochromatic and so condensed onto the periphery. So that will change the firing frequency of nearly all of the promoters around it in 3D space. So I think you can see that every base pair in the genome is affecting the activity of all others to some degree, and hence the pan-genomic model. So to summarize, I've said that all of these regulatory motifs are just promoters. And each one is going to act as one or other type of motif depending on your particular focus. As GWAS results show, many base pairs in the genome affect the activity of others to some slight degree. So the pan-genomic model naturally includes widely distributed gene regulation. In the next segment, I'll describe a simple formula that enables us to predict the activity of any promoter in any cell. Thank you for your interest.